What's up, YouTube? Let me turn this down. Shit. Alright, I'm here to do a crash course on plant roots, not plant roots, plant tissues, animal tissues. We're gonna go into porinifera, which are porifera, bleh, which are sponges, and cnidaria, which are jellyfish and hydra and things like that. So, first we're gonna go over plant tissues. I'm going to use these lovely slides that my professor uploaded, and we're going to crash course over into this. So, what you need to know about plants, what makes them different from animals, is that they have intermediate growth. What that means is that they never stop growing. That's why, trees grow for hundreds of years. Whereas plants, once we hit, not plants, fucking animals, once we hit a certain age, we stop growing. We're not growing any more limbs. We're not growing any new organs. Plants are always growing new leaves, new stems, new branches, new roots, and those qualify as organs to those plants. Now, one thing you need to know is where this indeterminate growth is coming from are these meristems. Think of them like stem cells for animals. These are undifferentiated cells. That means they're not specialized. They're just blank cells ready to be anything, ready to be a root, a branch, a leaf, anything. Meristems. It's like stem cells for animals. Now there's the apical meristem and there's the lateral meristems. Apical, think about vertical. So there's two apical meristems. There's one at the root, which is called the root apical meristem, and one at the top of the plant, which is called the leaf primordia or primordia. Let me go to it real quick so you can see how it's spelled. Well, that's what it looks like right there. Pretty sure it's right here. Now, chapter 9 has a lot of things, but chapter 10, it's a breeze, and I'm pretty sure the sponges and all that thing, it's a breeze too. Oh, it's called the shoot apex. The leaf primordia, I'm wrong, is what protects the meristem right here. The leaf primordia. It's like the root cap that protects the meristem at the tip of the root. So that would be, the root apical meristem. And at the top of the stem, you can call it the shoot apex. Alright? Or shoot apical meristem, okay? This is what it's called. Shoot apical meristem, I'm sorry. And at the root, it's called the root apical meristem, okay? Those are the two apical meristems, so the root apical those are the two apical meristems, I'm sorry. The root apical meristem, it has to do with growing below the bottom of soil. And the shoot apical meristem has to do with growing high, growing up, So, lateral meristem, lateral, think sides. So that means the plant growing fatter, growing wider, okay, growing thicker. And there's two for the lateral. It's called the vascular cambium and the court cambium. Vascular cambium has to do with making the stem thicker. Court cambium has to do with growing the bark, like the outer protection of the plant. Now, when it comes to there's two ways to classify plant parts of the plant. There's the roots, anything that grows below the ground, and shoots, the part that grow above the ground. All right. That's leaves and stems. Now, if in case he asked this question, which parts, what are the vegetative parts of the plant? That means what parts of the plant are not involved in sexual reproduction. So that would be the roots, the stems, and the leaves. Now, when it comes to plants, there's two classifications. There's monocots and dicots. The reason why they're called monocots is because they only have one cotyledon, cotyledon. And then dicot, there's two cotyledons, okay, in the seed. Otherwise, these are really important distinctions you need to memorize. Monocots, they have fibrous roots. That means the roots are scattered everywhere, but they're the same size and about the same shape and the same length. Vascular, that means the xylem and the phloem are arranged randomly. They're not organized. Let me show you an example of that right here. Right here. There's like no organization whatsoever in the vascular arrangement of the stem. This is a cross section of a stem. There's no arrangement. Whereas in a dicot, they're right here in like a perfect ring. Not perfect, like but you can tell it's a circle, okay? And that's what it shows right here too. Circle for dicot, random for monocot. Now when it comes to the leaves, the monocot leaves are parallel. That means they're never touching. It's like patterns, you know, like lines. Like think of it on this keyboard, like these lines never touching. Dicot, the veins on the leaves, they're like veins. I just said that, yeah. They're like veins, okay? They're like branching out. They're like a net, like it says right there. Now, monocot flower, think multiples of 3. Dicot, 4 or 5. So monocot, think of it mono, so it's the lowest one, so 3. Dicot, 4 and 5. 4 and 5, they're greater than 3. But don't just think of it that way. So it's just multiples of 3. So that means a flower, if it's 3 petals, 6 petals, 9 petals, it's a monocot. If it's anything else, dicot, okay? Now this is when it comes to angiosperms, you know, whatever. Now, plants have a root system below ground. So the point of the roots is to anchor the plant. Remember that, the purpose of the roots, anchor the plant to the soil. And the root absorbs water and minerals from the soil. And the water goes from the roots all the way up through the plant with the minerals. Well, that's pretty basic, you know, stuff about plants. So we've already touched on the differences. So monocots, fibrous roots. Oh, I never really explained dicot roots though. So dicot roots, there's a tap root, so that means there's a huge central root. Think of like a carrot growing on the ground. Carrots are actually roots. And then there's little roots coming out of the side, but there's this huge one that you can tell the difference. Shit, undo. Okay, continue forward. Now, in the root, it's different for roots in monocots and dicots. So monocots, there's a ring right here. In dicots, there's a little circle with like an X pattern on it. It's different from the stem, where in the stem the dicot has the ring. But the reason why you know this is a monocot root and not a dicot stem is because of this border right here called the endodermis. Remember that the endodermis you can see in roots, in stems. You really can't see an endodermis. Now, you need to know the different parts of the root cross section. So the outermost layer, epidermis. Between the endodermis, this second circle in the middle, and let me get a pencil. And between this endodermis and this epidermis, the section right here is called the cortex. And the bigger circles are the xylem, and the little circles right there, those are the phloem. This center section right here is called the pith. Alright? And you can basically call this whole area right here the vascular cylinder. or you can call it the style. Not the style. It's called something else. Stele. It's called the stele. Stele is S-T-E-L-E. Stele. I'll write it down right here randomly. I don't care. So let me go to the page just to make sure though. So this right here, this stele. He's probably not gonna ask this, but whatever, you know. Stele. S-T-E-L-E, whatever. Alright. It's the same concept with the dicot. You know, this border right here, it's called the endodermis. And the xylem is the big part of the X. Everything else, consider it the phloem. Alright. So this is still the cortex right here, this part right here. Epidermis, endodermis right here, this smaller circle. Alright. Yeah, there it is. Stele. That's what it's called. So stele basically it's the xylem and phloem, okay? Now, one thing to know is if you see in the lab exam that the border's pink, that border is called the Casparian strip. The purpose of that is so when water goes into the roots, it's being regulated by the Casparian strip, which is like a gasket. It's watertight. The only way water can get through is through this through the cells around the Casparian strip in the endodermis. All right. That's how roots regulate how much water they get in. Because if the roots need water, the cells will have more solutes than outside of the roots. That's how the water's regulated, so plants don't drink up too much and explode or something like that. Now, on the roots, there's these things called root hairs. You need to know what they do. Root hairs grow outside of the epidermis, and they're extensions from the epidermis, and their basic function is to increase the surface area so that the plant absorbs more water. It's basically to cover more ground. Alright? Now, in the apical meristem, there's different zones. You need to know about these different zones. I'm pretty sure there's a picture of it right here. If not, then whatever. But just in case, I don't see it. But in case he does ask. So you can tell this part right here is the root cap right there. And then you can say like this part right here where it's really, really, really detailed with dots. That's the zone of cell division. The reason why the cells are so small is because they're constantly dividing. And as they get larger, you can say, oh, this is a zone of elongation. And then as they get even more high up, zone of maturation. And that's where root hairs come from. Because that's where cells specialize. It kind of makes sense like that. Alright? So right here, outside here, root cap. Right here is where the meristem starts. And that's zone of cell division. Now these are important slides you need to be able to identify. When you see this, you need to be able to say, those are root hairs. When you see this really dark red border, that's so you know, okay, that's endodermis or Casparian strip. It depends on how he words the question. But since you see these xylem arranged in a ring, and there's an endodermis that's really obvious to see, that's how you know it's a monocot. In case he asks you what kind of plant it is, it's a monocot. Right here, cortex, pith, these larger circles, xylem. Smaller circles, phloem. Epidermis, outer layer, okay? This is how you know it's a dicot. There's an X. There's no other X in anywhere, like stems or roots. No, it's only dicots, roots. X. large circles, the large pink circles, xylem. These smaller blue circles around, phloem. Alright? And everything else around, cortex. Because this is the endodermis right here. So yeah, it says right here, be able to identify the following structures: dicot roots, epidermis, cortex, endodermis, xylem, and phloem. Monocot roots: epidermis, cortex, pith, endodermis, xylem, and phloem. Alright. So the shoot system, you basically might need to know how to label the parts of this stem right here, like this branch or twig. So just carefully study these things right here. Remember, lenticels, those are little spots in the opening of the bark so the plant can get more oxygen. Terminal bud scale scars, that's basically the plant eventually stopped growing, like here on this bud scale, terminal bud, but then because it was winter or something, and then it started to grow again, and that's why there's this mark right here, that little mark. You see it on branches everywhere. Alright, bundle scars, I don't know, I'm guessing that's where like it lost a leaf. No, that would be the leaf scar, never mind. Bundle scars, I'm guessing that's where the plant was cut or something like that. Okay. What's more important to know though is nodes, internodes. Okay, nodes is basically the point where a leaf are, the leaves are. I mean to say. So node, it's kind of like measuring a distance. So steps. And the distance in between, it's called an internode. So step, internode, node, internode. But the part where the leaf connects to the stem is called the petiole, which is P-E-T-I-O-L-E. That's pretty much it. I mean, you can start. Oh, and remember the angle that the leaf makes with the stem? It's called the axil. Like that. Axil. A-X-I-L. And an axillary bud is above the leaf where it connects. So you see how there's a leaf scar here? That means there used to be a leaf here. It got torn off. There's an axillary bud right here. That's basically if the leaf gets torn off, another leaf is going to grow there. Alright? And remember, plant's vascular system transports sugars from the leaves to the roots. Because the leaves make the sugars because they go through photosynthesis. Stem, this function basically support and transportation of water and sugars and all that. So yeah, the spaces between nodes are internodes. Leaf attached to stems at nodes, okay? This slide makes it so much easier. Terminal buds contain undifferentiated tissue at the shoot tip. Terminal buds, that's where the stem stopped growing. That's where the meristem is at. It's ready to grow into more, grow taller, since plants never stop growing. Each node contains an axillary bud where a new shoot grows out of. Alright? And they're always above where the leaf attaches to the stem right here. These are axillary buds. It's been 19 minutes. I'm going to have to cut this into sections. Like, holy... monocots and dicots have different arrangements of vascular tissue. I've already went over that. So at the stem, the monocots, they're all over the place, scattered. Dicots, they're in a perfect ring. Alright? So, this is the pith. Cortex is the area between the arrangement of this and the epidermis. Pith is this huge space in the middle. Now for monocots, you don't need to know about, you know, pith and cortex and all that. You just need to know this is the vascular bundles right here, these little random circles, and this is the epidermis, and this is the ground tissue. Dicots, yeah, you need to know these circles here in this bigger, you know, circle. These are vascular bundles. Here, this area in the middle is pith. Here's the cortex. Here's the epidermis. All right. And it says right here, dicots, vascular bundles are arranged in a ring near the epidermis. Monocots, vascular bundles scattered throughout the stem. No pattern in monocots. And these are more slides. He's probably going to use these on the exam. But just make sure you know the plain difference in case he uses different pictures to throw you off. And be able to identify the following structures: dicot stem, epidermis, pith, fiber cap, xylem and phloem; monocot roots, epidermis, fibrous sheath, xylem and phloem. Ooh. And this is the leaf prime, the apical meristem right here at the tip of a growing shoot. So this is at the top of a shoot. Think of these horns. These horns are newly forming leaves. This area right here in the middle, shoot apical meristem. In case he does ask this, yeah, this is vascular tissue right here. This blue area right here. I don't think he's gonna ask that, but he might ask this. What's this little bulge right here? This is the axillary bud. This is where a new shoot might grow out of too. This angle right here is called the axil. That's what the angle is called. That's pretty much it.